Hi everyone, it's Mr. Sinti, and today I have the pleasure to talk to you about transportation in plants. And as we're going to see in this video, there's three main ways in which plants can transport materials like water and other dissolved minerals and sugar. And that is cell to cell through diffusion and osmosis, and then sort of short distance transport between tissue. And then there's something called bulk transport, where the plants are able to really drive a large quantity of water, like for example in trees, through their vascular tissue. And so let's jump right into that conversation. And so one of the first things that I want to say about that is that, do you see this picture here? This is a really neat picture of brown algae, which is found at Monterey Bay Aquarium here in California. One of the things about algal plants, they're not really true plants. Algal species are part of protists, and so it's a type of brown algae. And one of the things about this is that, when you live in the water like this, there's really no worries in terms of where am I going to get my water. It's like when you're a terrestrial plant, I think that's the biggest worry of all that plants have, is that running out of water, dehydrating. But you got plenty of water, and like, well, where are you going to get your minerals? Well, all of that's dissolved in the water. And you're like, well, where am I going to get my carbon dioxide for photosynthesis? Again, that's dissolved in the water. And then what about light? Light comes down and penetrates the water. Obviously, they can't live very deep, and so they have to be in the photic zone. But there's really no worries. But then when plant-like ancestors decided to colonize the land, they needed to evolve sort of that evolutionary journey. They needed to evolve sort of a differentiation in body plan. In other words, shoots or a big trunk like you see here in this redwood tree, and a root system, which are specialized areas to obtaining the things that terrestrial plants need. Like, for example, the shoot system is designed to capture, let me sort of just illustrate this. The shoot system is designed to capture sunlight up here in the leaves, and also carbon dioxide comes in up here in the shoot system. And then in the root, you have the ability to take up mineral elements and water. And so it's sort of differentiated this way. But in order for the shoot and the root to sort of coexist, there must be a way to transport. Do you follow that? Like, in other words, if sugar is being produced up here in the plant, in the needles, the sugar's got to be able to come down and be able to provide nutrient for the root system. Likewise, water and minerals like magnesium or potassium or sodium or iron, those, if they're being taken up in the root system, they need to be able to travel up here to the top of the tree. So there needs to be some cooperation. And so thus the evolution of vascular tissue. And so this is I think it's a really cool conversation about how the shoot and the root sort of, think of it, you could just sort of talk about vascular tissue, or you could think of it as sort of a way in which the shoots and the roots are able to work together to accomplish their goals. I like that a little bit better. And so one of the first things I'm going to say is these trees can get really tall. And so one of the things is, how is it that water can defy gravity and go up this high up in a tree? That's one question I have. But the whole idea of vascular plants, in other words, plants that have vascular tissue, well, the word vascular comes from vasculum, which is probably mispronouncing that in Latin. So vascular plant, and then they're known as tracheophytes. I like this, tracheophytes. Phyte means plant. Tracheo is from a Greek meaning trachea, sort of like a tube that your air passes through in our body. So these are tubes that conduct material up and down, sort of bulk in the tree. Now, as opposed to what are not tracheophytes? What are not tracheophytes are bryophytes, which are moss. Now, no slight against moss. Again, one's not better than the other, but without vascular tissue, notice the tree, how tall it can grow. Moss, you never see it growing tall. It always hugs the ground. It's even hugging a tree right here. And so what's interesting is that it gets all of its water and minerals from the soil, and it has to just simply diffuse. It can't get any taller, or else um, it wouldn't be able to survive. And so bryophyte, tracheophyte, kind of interesting. So one of the things that we're talking about here is sort of the solution, morphologically structural solution to solve this transport between roots and shoots is this vascular tissue. As opposed to what? Dermal tissue, which is on the outside for protection and absorption. Then you have this cortex material. Can you recognize this? This is a cross-section of a, look closely at the stele, which is this vascular tissue, this ring in the center. This is a monocot, correct. And so what we're talking about is this vascular tissue allows the plant to be able to move large quantities of material from the root, where it's absorbed, all the way up the stem. And water is needed up on the top for photosynthesis. And so the transportation takes place on 3 levels, and we're going to have that conversation today about how cells uptake water and solutes. And you might be familiar with this already. It's called diffusion and osmosis, from cell to cell, like that. But then we're also going to talk about briefly short-term or short-distance transport. Now, short-term is like on the level of the tissue. So this is a cell to cell, and this is tissue. What do I mean tissue? So, in other words, let me draw a root here, right there. So if water is going to enter into the root here, that's number 1. In other words, from soil to cell. And then if water is going to move between cell to cell like this, this is between tissue. That's number 2. This is short transport. And then once you get into the elevator system, in other words, once you get into the xylem, then you're going to be able to really cruise. That's long distance transport, and that's either within the xylem or the phloem sap. And so let me illustrate that. Come over here for a second. I think you're familiar with this concept, but let me just make sure. If you were to consider, for example, let's look at this. This is a cool little video clip right here. And so you can see in the root system here, water is taken in. And then what happens is once it enters into the vascular tissue, this is a picture of a tracheid right here. And notice the water molecules. They're all stuck together. Do you recall that water is very polar, and so polar molecules attract each other through hydrogen bonding? And so this is a great example of cohesion. So it's not only cohesion, but it's adhesion against the walls, the cellulose walls of those tracheids. So you go all the way up into the leaf and look at these guard cells here in that opening called the stomata. This is where water is going to exit or transpire. And so as each water molecule moves and evaporates, it's going to provide a tug, and so it pulls the water up the xylem. And so here it is. Let me pause this for a second. So we have osmosis taking the water in the epidermal cells, and then they move laterally across the tissue of the cortex, and then they move into the vascular tissue. So there's 3 kinds of transport going on. So that's pretty cool. And then along those lines, let's take a look at this. Same sort of thing is happening here. You could see first water is entering down below in the root, and then it's not only water, but it's dissolved minerals as well, and then that enters into the vascular tissue, and it's moving all the way up the tree. And so the reason that water can do that and defy gravity, I mean, gravity's trying to pull things down. So you've got a combination of cohesion between water molecules, and you got a little adhesion occurring as well due to the polarity caused by the cellulose and the water. So both of those together are known as capillary action. And so capillary action will cause the water to move on all the way up like that. And then up here in the leaf, there's vascular tissue there in the vein. And then simply the water since it's connected, will move across into the mesophyll, and then ultimately it will transpire through the guard cells, and this provides a pull. So the real reason water moves up a plant is that it's being pulled up through transpiration. And it's like, well, what's causing that? Well, there's low water potential in the atmosphere. There's a lot more water potential in the plant itself. Also, heat will cause water to evaporate, and also wind will increase that as well. And so that's pretty neat. I think you're familiar with most of those ideas. And so let's consider this conversation back to where we were in the notes. And so as you can see in this diagram, notice here that the light is creating the transpirational pull. But then, you know what's really interesting here is take a look down below in the soil. Do you notice here it's kind of weird? Is that oxygen is being taken in down here in the soil? Well, you got to expect that because these are living tissues, and it's like these cells need oxygen. You're like, well, where are they going to get it? Up here there's no problem because oxygen is produced by photosynthesis, but roots need oxygen. And one of the concerns that trees have, like for example, really popular trees like a redwood grove where there's millions of visitors that come each year to see these trees. Sometimes when people, let me illustrate this, say a person were to come like this, and you think like you're not really doing any damage, but what you're doing is you're flattening the soil right there and making it sort of impermeable. And so the more people that come and come and come like this, it sort of hardens the soil, and then oxygen isn't able to diffuse or percolate into the soil, and sometimes the root system can suffocate. Likewise, water isn't able to penetrate the soil either. So that's kind of an interesting thing. Just wanted to point that out. So this conversation might ring familiar. It's about how the molecules move across cell membranes, because cell membranes are basically the way in which not only plant cells are selectively permeable, but really all cells. And so the cell membrane is very discriminating. When we say semi-permeable, it means sort of half things can come in, half things cannot. What determines permeability by far is the phospholipid bilayer. The phospholipid bilayer is this lipid barrier that's going to prevent anything that any ion is not going to be able to pass through the phospholipid bilayer. And anything kind of large isn't going to be able to go through, or anything large and polar are not going to go through. But we do have, as you recall, these embedded integral proteins that sort of bisect the phospholipid bilayer, and those are called transport proteins. And those are able to move materials across very selectively. And so let's take a look at that. And so, for example, do you notice here these sort of like yellowish orange squares? These look like they're impermeable to the cell membrane. Maybe they're some kind of ion. And so each transport protein is very specific. It's sort of like an enzyme that way. It's very particular to a substrate. You can even call them permeases because they're very selective. And so things generally move passively from an area of high concentration to low, and that's simply diffusion. Diffusion, again, sometimes people think that molecules have a mind and they're like, I'm supposed to go from high to low. Molecules don't move literally from high to low. They do overall, but molecules just randomly move. So all things are in motion. So if molecules are in random motion, if more of them are on one side, over a period of time, they'll probably equal themselves out, if that makes sense to you. So we could say that molecules move from an area of high concentration to low. But like, let's put some examples to this. Like, for example, say that these transport proteins are very selective. And so the membranes of plants might have potassium channels. This is considered to be a channel that allows only potassium, which is a cation, to pass. But even something that's also a cation, like sodium, cannot. And so these doors, if you will, are specific. So if this is a sodium right there, if that's a sodium channel right there, and this is sodium, that means that sodium is going to be the only one moving through, and potassium, so sorry, you can't get through. Or hydrogen, so sorry, you can't get through. And so these are very selective channels. That's important. And so not only are these permeases selective, but some of them can even do some cool things. They can even move materials against the concentration gradient. And I'll give you some examples of why that might be the case, but let me just establish what this means. And so Active transport is, I'm not going to draw the whole phospholipid bilayer, but let's just say that this is the phospholipid bilayer, and here's a transport protein. And let's say that the concentration over here of whatever the substance is very, very high, and over here on this side is the concentration is very low. And so normal passive diffusion would suggest, let's go X right here. And so if it's very high, there's a lot of it, like that. So passive diffusion would suggest that molecules would, though that they can move back and forth, more molecules over time would move from where they are to where they're not. Whoops, I'm sorry, I meant to put low on this one. We go like that. And so molecules move from high concentration to low. So what's active transport? Active transport is going in a reverse direction. And so it requires energy to do that. Like say, for example, the cell over here, say this is the inside of the cell, right here. Say the cell wanted to get rid of X, even though there's a lot of X already present on the outside. The cell could do that. It would need to require energy, and energy comes from this molecule called adenosine triphosphate. You are familiar with it. Adenosine triphosphate, what can happen is the cell uses that chemical currency by hydrolyzing the third phosphate group from it, and it removes it. And as a result, it produces adenosine diphosphate, and the third phosphate can stick onto a protein, and because it's a negatively charged functional group, it causes a protein conformational change which can scoot molecules against the concentration gradient. And so, check that out. A molecule can actually move from an area of low concentration to high with the help of ATP. Pretty neat. We call that active transport because it requires energy in the form of ATP. Now, what's an important example of this? I can think of no more important active transport than when plant cells produce this active transport when they're working with protons. And so it's called a proton pump. A pump implies that you're pushing something. And so what the cell does is it tries to move protons from an area of low concentration to an already high concentration. And so the most important active transporter in the plasma membrane of plants is the proton pump, and it needs ATP to do that. And so, now why does it do that? Why does it pump protons from low concentration to high? Well, this is the reason. It wants to create a membrane potential or a voltage by doing that. And so, check this out. Let me sort of take a break from the notes for a second and sort of illustrate this. And so let's go here and take a look. Let me see if I can do that. I'll make a blackboard like this. Okay, let's say that you're a plant cell. Let's say you're a root cell epidermis. This is the root hair, like this. We do another one over here and another one over here. So what's the story? This is inside the root. This would be dermal, this would be ground over here, this would be the cortex, this is the soil over here, and then we have protons like this in the soil, and we have them in the cell. So what's the story here? So what the cell can do, and it's surprising because sometimes I don't know if you consider the roots to be alive, but they are. And these cells are alive and they do cool things. Look at this. They can put membrane proteins that are specific for protons. And check this out. You know what they do? They use this and they take these protons and they push them out here and push it out here and push it out here. Notice how the concentration in the soil is very high with protons, and it's going to be relatively low on the inside. And it's like, how can it push it? It's pushing it because the cell uses ATP in order to drive that against the concentration gradient. And so why does it do that? Well, it does that. didn't really mean to erase that. It does that because when it pumps all those protons to the soil like this, all these protons are out there, ultimately what's going to happen if there's more protons on the outside, it's going to make it relatively positive in the soil and relatively negative in the cell. And so that is what we call in biology a voltage gradient or a membrane potential. It's a voltage gradient, meaning like there's plus on outside and negative on the inside, and it's a membrane potential. Because now, check this out, now any sort of cation like calcium or potassium or sodium, any of these things will be more attracted to come in to the cell now that the root has created this not only membrane potential, but these ions are attracted to the electricity. And so what you've created is a electrochemical gradient. So not only are the hydrogens in high concentration and they want to diffuse back. They're very uptight about that. So there's a chemical gradient and a voltage gradient. So it's an electrochemical gradient, and the root takes a lot of effort to do that. Now, why does it do that? Why does the cell create that? Well, I mentioned it sort of helps to move things in. So here's a picture of what I was trying to draw. Say this is the inside of a root cell. So what the plant will do is it'll push protons, push them out. Look, it requires energy. It's pushing them, even though it's high concentration, active transport. What is this called? A proton pump. It pushes and pushes, and it creates an electrochemical gradient. In other words, positive out here, negative here, and high concentration of protons, low concentration of protons. So it's a electrochemical gradient. Now, why does it do that? Why does it work so hard to do that? Well, an example of this would be, I mentioned this, that once the electrochemical gradient has been established, one of the favorite ions of a plant is potassium. Sort of like we like to eat potassium too, like for example in bananas have a lot of potassium, fruit in general. So we need that. And so check this out. Cations, for example potassium, are then drawn in. Now, they don't go through the same proton pump, but they go through their own potassium transport protein, but they're more readily drawn into a root because they're attracted to the negative charge. So the electrochemical gradient helps cation uptake. That's the answer, helps cation uptake. So more examples. If it's crucially important, there must be even more, and there are more examples. So check this out. I find this very, very fascinating. So watch this. So after the root pumps out all these protons out here, check this out. It can even help negative ions such as nitrate. Now you know through our study of the nitrogen cycle, taking up nitrogen is absolutely critical. And you're like, well, whoa, if you make it negative on the inside, then the nitrate is not going to want to come in. But check this out. Sometimes these proton gradients also help in what are called co-transports. So do you know, like, here's an analogy. So you're going into a big hotel and there's a door, those glass doors that sort of revolve to get into the hotel. Do you know how, like, sometimes it's fun with you and your friend to both go into the same little pie, and then you can both be transported in at the same time? Check this out. There's some co-transports that will allow protons to get in. Now, why would protons be going in? Because they're attracted to go inside the cell because of what we said before. Do you remember we said that protons, things that are positive, cation uptake is increased? Well, check this out. Over here, you're pushing protons out, pushing it out, but then this door will allow protons to come back in. Of course, they want to come back in. But not only do they come in, but they also bring in anions with them, because two people, or two solutes, can get into this transport at the same time. Isn't that neat? So you can take in anions as well by creating this electrochemical gradient. And if that's not enough, this is another huge one. Check this out. Here's an example of, it looks the same. It's co-transport, but this time it's a neutral substance. So check this out. So the protons go into this co-transport, and then something neutral like sucrose, which is a disaccharide, can sort of buddy up with the protons and then be shuttled into a cell. And you're like, okay, now what is up with that? And so let me give an example of that. An example of that is, let me come over here. So an example of that is, okay, here's a cell right here. And let's say that that's in the leaf. And say it's a mesophyll cell and it's making lots of sucrose. Well, it makes GAP, and then it turns that into glucose, and then that can be turned into sucrose. Okay, so there's sucrose, and it's building up inside the mesophyll, and you're like, well, what I would like to do is put the sucrose into the vein. And it's like, okay, so we got to put it in particular in the phloem. Do you remember that phloem is specialized tissue that will take sugar water? And so we're like, okay, so I want to get this into the phloem. So here's the phloem cell. Do you remember the phloem cell is called a sieve tube member? And it's like, how are we going to do that? Well, check this out. The phloem cell can do this. It can pump protons. These are protons. It can pump protons over here like this. Pump protons over here. Now, why would it do that? It's pumping it, pumping it, pumping it. So now this is relatively positive. This is relatively negative in the sieve tube. So then over here, check this out. This is one of those co-transports. Let me just sort of make it cool looking if I can. And the reason that it's a co-transport is that it will allow not only sucrose to go in there, I'm not going to change color, but a proton. And so it'll shuttle sucrose into, this is a good way to get a neutral molecule into the sieve tube. So you can get lots of sucrose in there. So then check this out. So this is like a powder. You know, like, well, wait a minute. Like if this is getting very sugary inside the phloem. We remember this term? It's called hypertonic. If the sieve tube is becoming very hypertonic, then watch this. Adjacent to, I'll keep the same color, adjacent to the phloem cell is a xylem cell. Now xylem has water moving up, right? So here's all that water and water and water like this. And so water is going to look over here and say, hey, 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 look, it's very sugary over here. It's very hypertonic. And so water, watch this, is going to diffuse through osmosis from an area of high water potential to low water potential. And the low water potential is caused by the increase in solute. So the water moves over like this. So it's like this. It's like the water's rushing in. And so you ready for this? This provides a pressure, water, to push the sugar sap down the phloem. How do you like that? And so phloem sap is pushed by pressure that was generated by the movement of water into the phloem. And then it's like, well, wait a minute. If this gets building up, so down here, the way this is maintained is down here in the phloem is that the sugar is removed. Like say, for example, this is the root. Now, this is a very small plant. This is the leaf up here. So if the sugar is offloaded down here in the root, check this out. It'll always be a situation where the water is flowing in as a result of the co-transport, which has been generated by the proton pump. I hope you're enjoying that. I think that's one of the cooler things about our conversation that we're having today. And so what I wanted to say about that is that phloem sap, this is what I was trying to explain, when it was being measured, and that's a whole nother discussion of how phloem sap can be measured, the rate of it, in other words, how much sap is moving like per hour. the rate was faster than can be accounted for by just simple diffusion between cells. There had to be something sort of pushing it. And so phloem sap moves by bulk flow driven by pressure. And the reason is that these higher levels of sugar at the source, in other words, in the sieve tube, lowers the water potential, which causes water to come in from the xylem. And then that sort of difference in hydrostatic pressure drives the phloem sap down from where it is in the leaf down to the sink, which will be the root. And so this is what I was trying to draw here. So because of the co-transport, sucrose is put in here, and so it's kind of sugary, which then allows water to cruise over from the xylem, which then pushes the sap in a downward direction like that, and then the sugar is offloaded. Do you see that? So it's constantly, the water's flowing, mostly here because it's hypertonic, and then since the sugar is offloaded down over here, like this looks, for example, like a beet. It's very sugary, sugar beet. Then the water then goes back over here, and it forms like this loop, like that. Pretty cool. And so finally what I wanted to say is these proton pumps, I don't want to confuse you, and I think the mere mortal student can be confused. This is sort of opposite of what we have talked about before in the mitochondria and in the chloroplasts, when those two organelles use an electrochemical gradient to produce ATP. What I've been talking about is the use of ATP to make an electrochemical gradient. Now it's just the opposite. The energy's got to come from somewhere, and so as you recall, in the mitochondria, the energy comes from the oxidation of the food that we eat, and that creates these redox reactions, which creates an electrochemical gradient. Let me just briefly, I won't go into it too far, but in the mitochondria, like this, what the mitochondria is capable of doing is using the reduced coenzymes in the Krebs cycle, which occurs in the matrix, and it uses an electron transport chain to then put protons out here, and then it uses this proton gradient to then allow protons to move back in to make ATP in what's known as chemiosmosis. So here we're trying to make it. Same thing's happening in the chloroplast. Like, for example, this is positive in the inside of the thylakoid and negative in the stroma, and that's created by the electrochemical gradient. And so this is how the plant is able to make ATP, and that's used to produce sugar in the Calvin cycle. So pretty awesome. It's hard to imagine that protons can do so many cool things. And so I hope you enjoyed that conversation of transport in plants. Thanks for watching.